Our next speaker is Dr. Gideon Blumenthal, who is a clinical team leader of thoracic and head and neck oncology at the US FDA. He did his medical oncology training at the National Cancer Institute in DC, and he continues to do some clinical work there as well. But he has personally been presenting some new work that the FDA has been looking at in terms of the endpoints that we have traditionally used in lung cancer, and trying to reframe how we might approach clinical trial development for this new molecular oncology era. So I'm very happy to have Dr. Blumenthal here to speak. I'm sure that you are all, including the faculty, interested in FDA perspective on how we are going to develop these new drugs to get them to patients in a timely way, but still be very thoughtful about how to attend to safety and efficacy of these drugs. Thank you.
Thank you, Jack, for inviting me here. I'm equally happy to be here with all of you, the patient community, the thought leaders and academics in lung cancer. Thank you so much. So the usual disclaimer, I don't speak for the FDA. It's a big institution. These opinions expressed are my own. So I wanted to just give a brief outline of the presentation. I think it's important just to briefly touch upon my interpretation of FDA history in a nutshell, and just give an overview of expedited programs that we have in place. Also, I wanted to talk a little bit about lung cancer, where we've been in the past 20 years, highlight some of the targeted approvals that you all are very familiar with, and then move on sort of forward-looking into novel approaches and trial designs. And finally, I'll touch upon, just briefly, an effort within FDA is we really want to incorporate more the patient voice into drug development, as the patient is the end user. We all are going to be patients one day, and it's critical that patients have a seat at the table for these critical decisions. So this is my highly condensed FDA timeline. Someone alluded to earlier, I think Dr. Weiss, that in Washington, people like to kick the can down the road, and I think that's very astute. The history of the FDA is actually, and the authorities provided to the FDA are very reactionary, and they're based on various health crises that have occurred over the past century. So starting in 1906, Teddy Roosevelt signed the Pure Food and Drug Act. He was outraged by the muckraking, the practices in slaughterhouses uncovered by muckraking journalists like Upton Sinclair, as profiled in the book The Jungle. So there was a requirement to properly label food and drugs. Moving on to 1938, the Food, Drug, and Cosmetic Act was in part prompted by diethylene glycol found in sulfonilamide, which was used as an anti-infective agent, and there were hundreds of deaths. So instituted was a requirement that food and drugs must be safe prior to marketing. 1962, there's a picture of President John F. Kennedy with Frances Kelsey. She was awarded the Presidential Medal of Honor. She was an FDA reviewer, and she did not approve the shipment of thalidomide in the United States. Thalidomide was given widespread in Europe as a sedative to pregnant women, and led to thousands of serious birth defects. So as these Kefauver-Harris amendments to the Food, Drug, and Cosmetic Acts enacted the safety and efficacy requirements, so it was incumbent upon sponsors to prove that the drug was efficacious prior to marketing authorization. This also enacted modern regulations into clinical trials. Moving on to 1983, I have a picture of Annie here. This was the Orphan Drug Act. This was critical for study of rare diseases. It incentivized the industry to study these rare populations, very important in this era of molecular oncology. 1992, as part of the AIDS crisis, many patient advocacy within the HIV community advocated for more rapid approval of drugs, and the accelerated approval regulations came into being. 2007, we have Vioxx, and as a reaction to that, the FDA was given new authorities to regulate the safety of drugs in the post-marketing setting. And finally, in 2012, our most recent legislation was called FDASIA, and I have a picture of the doors, the break on through to the other side. This was where, among other things, the breakthrough designation was granted. I'll talk about that, as well as accelerated approval in subsequent slides. So this is just a summary of some of the key FDA expedited programs. These range from preclinical studies through early clinical studies to the pivotal studies to get marketing authorization, FDA review, and FDA approval. So they span the whole gamut of drug development. So what is a breakthrough therapy designation? These are for drugs which are intended alone or in combination to treat serious or life-threatening diseases, in which preliminary clinical evidence indicates that the drug may demonstrate substantial improvements over existing therapies. So in summary, they're really for transformative, sort of knock your socks off treatments. Oftentimes we get the question, how do you know if it's a breakthrough therapy? And it's a hard question. And it's sort of like it when you see it. It shouldn't be subtle. It should be a really transformative type therapy. Once breakthrough therapy is designated, an all-hands-on-deck approach ensues involving senior reviewers within the agency, senior managers, to work with the companies to try to get these drugs on the market as quickly as possible. Now, FDA giveth, FDA can taketh away. If with further study, the drug doesn't seem to be panning out as it initially had, breakthrough therapy designation can be rescinded. To my knowledge, it's never happened. It's still early on in the process. The other thing I should mention about breakthrough designation is what we found early on in the process is usually it's not the clinical data that's the rate-limiting step. It's oftentimes the chemistry and manufacturing. Can the sponsors scale up their manufacturing processes to ensure that they can give a safe and quality dose of the drug? So that's been some of the challenges that we're looking at internally, is how do our chemists and manufacturing experts work with the companies to facilitate scaling up of these processes, which are oftentimes challenging to go from early phase 1 studies to approval. We don't have that 10-year runway that we used to have. Now it's 1 or 2 years, very challenging. So there are two paths to approval. One is the regular approval, and that requires, it's in the Code of Federal Regulations, it requires substantial evidence from adequate and well-controlled trial or trials. In other disease areas like diabetes, high blood pressure, most often it requires several trials. In oncology, typically we take one trial because we realize that the risk-benefit is different, and patients need these drugs quickly, and so we pay more of a focus on the efficacy side, and we know that patients and doctors are more willing to tolerate toxicities. Regular approval is based on improvements in direct measurements of clinical benefit. So you have to demonstrate a longer life or a better life, how a patient feels, functions, or survives. Or it can be approved on an established surrogate. What is a surrogate endpoint? A surrogate endpoint is something that you measure, doesn't necessarily, it is not clinical benefit in and of itself, but if you measure a change in that endpoint, that signals that, that the patient, for example, will live longer. An example would be in HIV. You look at HIV viral load. It's been very well established that you give a drug to a patient, the viral load clears a certain threshold, that patient will have reduced complications, opportunistic infections, and will live longer. So that's an established surrogate. And in the HIV world, you clear a patient's viral load with a drug. That's a well-trodden path towards approval. Accelerated approval, on the other hand, is not approval light. It's not a lowering of the evidentiary standards. However, these are based on improvements in surrogate endpoints that are reasonably likely to predict clinical benefit over available therapy. So these are not as established surrogates. We think that if we see a large effect, for example, on response rates that are durable in patients, we think that this will translate. But we haven't quite established that same level of surrogacy as the HIV community has with viral load, for example. And with accelerated approval, the sponsors are required to conduct post-marketing studies to confirm clinical benefit. So from a regulatory standpoint, we're taking on some risk. We have more uncertainty with these endpoints, but we require that the studies be conducted in a post-marketing setting with due diligence. So just to highlight the lung cancer approvals in the last 20 years, up until 2011, all of our approvals were in unselected patients, not molecularly enriched. This is a list of frontline therapies, maintenance therapies, second line and beyond, and one drug in combination with photodynamic therapy was approved for symptoms of an obstructing tumor. And as you are well aware, recently there have been a number of approvals for targeted therapies in molecularly enriched populations. The first to come about was crizotinib in ALK-positive non-small cell lung cancer. The accelerated approval occurred in 2011. This was 4 years after the ALK fusion was first described to be a driver oncogene in lung cancer. And 2 years later, just last year, it was converted to regular approval based on the randomized study that you've seen already showing improved response rates and progression-free survival against chemotherapy. Erlotinib and afatinib are both approved in L858R and deletion 19 EGFR mutation positive lung cancer, both based on improved response and progression-free survival against chemotherapy. And most recently, I'll walk you through this in the next slide, ceritinib was approved for patients who progress on crizotinib. And ceritinib was the first lung cancer drug approved in the Breakthrough Designation Program. So this is, I hope, a good example of what the new paradigm is for drug development. Ceritinib entered first-in-human studies in 2011 based on very exciting response rates and durability. It got breakthrough therapy designation in March 2013. In November of 2013, the new drug application was submitted. It was given a priority review, so the typical timeline for a priority review is 8 months. However, we worked really hard with a multidisciplinary team of statisticians, pharmacologists, toxicologists, chemists to approve the drug in April 2014 in patients who had progressed on crizotinib. So less than 3 years, or rather a little over 3 years from first-in-human studies to FDA approval. Certainly much shorter than the typical decade that it might take to develop a drug. So moving on to novel designs and endpoints. So as Jack alluded to, this is a schematic of a typical randomized control study versus a single-arm study. And the template for decades in lung cancer had been to take a very heterogeneous patient population. We didn't have the great tools we have now to sequence the cancer genome very quickly and cost-effectively. And patients in these heterogeneous group of patients would be randomized to, say, platinum doublet versus platinum doublet plus drug X, look at longer-term outcomes like overall survival, progression-free survival. And the challenge with this approach is, as was seen for the past decade, there's a high risk of either a failure of the study to meet its primary endpoint, or it could be you could have a statistically significant event without, statistically significant outcome without having necessarily a clinically meaningful outcome. Or you could have a subset of these patients really benefiting from the drug and some not, but without characterizing these patients molecularly before entering them on study, it's hard to know who's driving what. And you're extrapolating results from a large population into really what may be only a small patient population that might benefit. And then we have single-arm studies. ALK would be a great example of smaller patient numbers, maybe 100 to 200 patients. They're identified with the genetic aberration from the get-go. They're given a targeted therapy, and the endpoint there would be overall response rate. So there are some pros and cons to single-arm trials. One of the pros would be smaller, faster trials. It's more feasible to enroll patients with rare tumor types. You can use response rates to look for large effect because you're confident that tumors in general don't spontaneously regress. So you know that that's a drug effect if you can shrink the tumor. The other thing that's important to note is if you see early activity in early clinical trials, is there really equipoise to randomize patients to what's thought to be a less effective chemotherapy control? So can we even ethically conduct randomized clinical trials if we see large treatment effects early on? There are some caveats, however, to single-arm trials. There's no randomized comparison. You could lead to bias in selecting patients. Maybe you're only enrolling the marathon runners into your trial to sort of jack up that response rate. You don't necessarily get great information on longer-term outcomes because you could be enrolling, those patients with more indolent disease. So you don't know if you're changing outcomes necessarily. And you also don't get that good comparative safety data that you would in a randomized study. This is some data that Jack alluded to earlier that we did at FDA. We looked back at all of the submissions that have come in within the last decade, 14 trials, over 12,000 patients. And we looked at on a trial level, differences in response rate versus differences in progression-free survival. And we actually saw a very good association between response rate and progression-free survival on a trial level. This suggests to us that if you have a drug, typically it's going to be in a molecularly targeted population with a very high response rate. If you compared that drug to chemotherapy, you might also expect a very large magnitude of progression-free survival benefit. We actually didn't see the same association with overall survival. However, there may be some caveats there which complicates the assessment of overall survival, like patients crossing over to the active treatment group after progression. So moving on to another sort of novel paradigm that we at FDA are very interested in, and we've been an active advocate in helping to facilitate these types of trials, are what's called the master protocol. We at FDA acknowledge, as Jack alluded to, in this new era of molecular oncology, in a single institution, it'll be impossible to find those 1% patients. So we're going to have to pool our resources, work together, build networks of clinical trialists, and form these networks to answer important questions. So I have the schematic of an umbrella protocol or a basket protocol. These are two sort of general outlines of what these master protocols might look like. An umbrella trial might test a drug or multiple drugs against a whole host of mutations in one single type of cancer. There are some examples. I'll have a slide on the lung map in a second. And then a basket study would be to test a drug or numerous drugs on a single mutation across a variety of cancer types. So if you had ALK-positive lung cancer or ALK-positive neuroblastoma, you could potentially be entered into the same trial. This is an example of the lung map, which we were very involved in early on in sort of guiding the process. This is a multi-stakeholder, the National Cancer Institute, the FDA, the Foundation for the NIH, all of the cooperative groups, pharma, the device manufacturers, all got together and said, let's design a single umbrella trial for second-line squamous lung cancer. Patients get broadly profiled with next-generation sequencing, and based on their tumor type, they get allocated to a number of sub-studies, randomized either to a targeted therapy or a standard control. And then there's a non-match arm. Patients who don't meet any of these criteria will get randomized to an immunotherapy versus a control. So the beauty here is you have pooling of patients, you have economies of scale. If you add or drop new drugs into the protocol, you don't have to go through all of the hurdles to initiate a new study. And this study is designed, if it meets certain criteria and shows favorable risk-benefit, potentially these drugs can file for FDA approval. So finally, my last few slides will be touch upon efforts to involve patients. In June of last year, we held a meeting, a patient-focused drug development meeting in lung cancer. And we want to hear perspectives from lung cancer patients about the disease, impacts on daily life, and currently available therapies. And this is part of a program, part of PDUFA 5 in Washington. In government, we have a lot of acronyms, so that's one of them, PDUFA. And you can read the report at the link that's provided in the slide deck. The other thing I want to briefly touch upon is patient-reported outcomes. It's incumbent upon all of us to develop better patient-reported outcomes and implement them in clinical studies, because we really want to, looking at these CT scans where the tumor, the lung is effaced by tumor and then it melts away with a targeted therapy, intrinsically we know that that patient must be feeling better, but we need to have better tools to capture that and implement them into studies. So just some parting thoughts. I think all of us as FDA, patients, physicians, drug and device manufacturers, we do share some mutual interests, and that's the rapid development of highly effective and safe medications for patients. I showed you some of the expedited programs that we have to facilitate the development of such therapies, but we need to do more. We need to develop novel endpoints, clinical trial designs, patient-reported outcomes in this new age. And all of you play a crucial role in guiding drug development. Thank you.